So today it was quite exciting. We saw that mitochondria, ROS are really key pathways for preconditioning. And with my talk today, I would like to give you a different view and put the emphasis on the ER, endoplasmic reticulum, which we think has also a major role in this preconditioning system. So in our lab, we are working in neurodegeneration and focusing on this unfolded protein response and autophagy, together with proteasome, are the main pathway which regulate protein homeostasis, also called proteostasis. So we have been focusing on the endoplasmic reticulum because it's a key organelle to regulate protein homeostasis. Interestingly, in neurological disease such as Alzheimer, Parkinson, ALS, or prion disease, which have all different clinical manifestations, they all share a similar accumulation of misfolded protein, or pathological misfolded protein. And the unfolded protein response is a key mechanism to regulate the load of those misfolded protein response. So here we have a view of this apparently complex pathway, but the result of this activation of the unfolded protein response is quite simple. First, it reduces the load of protein by reducing translation, so reduction of translation. Second, there are ER chaperones which are produced, which help with the folding. And third, you have the activation of the ERAD pathway, which help degrading via the proteasome misfolded proteins. So within this pathway, I will go a little bit through it because we're going to speak about mutants of this pathway. You have the IRE1 pathway, which is very well conserved from yeast to mammals, which is actually a peculiar protein because not only it has a kinase activity, it can autophosphorylate, but also it has this endoribonuclease activity. As you can see here, this endoribonuclease activity can undergo the splicing of XBP1, and when XBP1 is spliced, it goes to translocate to the nucleus to allow the expression of chaperones, activate the ERAD. The second important pathway is the PERK pathway here, and the PERK pathway is the one who can reduce translation. It can also induce the activation of a transcription factor, ATF4, which allows the expression of chaperones. And importantly, if this pathway is overactivated, it leads to the activation of a pro-apoptotic protein, SHOP, which lead to apoptosis. So meaning that if the load of protein or misfolded protein is too high, the ER will trigger apoptosis. The last pathway is ATF6, which allows the expression of chaperone, activation of the ERAD. So in the lab, basically what we ask is whether modulation of this pathway could affect the degeneration of neurons in pathological disease. And so basically we made the hypothesis that during neurodegenerative disease, you have the death, apoptotic death, it can be also necrotic death, and that endoplasmic reticulum and possibly autophagy may interfere with the death of the. To address this question, we use basically the simple model organism, which is Drosophila, because it has we can do powerful genetic screen, isolate mutants, basically that would induce ER stress and modulate the pathway. And one of the key advantages of Drosophila is that it has low genetic redundancy, it's cheap, can do drug screening, and also 70% of the genes involved in disease have a functional homologue in Drosophila. As a model, we use the Drosophila retina. Why? Because it's packed with neurons. It has been widely used as a model of neurodegeneration. And also it contains those neurons which are tightly associated with pigment, glial cells, so it's also a nice neuron to glia communication system. So using this model, we basically were able to screen many mutants, and I'm going to speak to tell you about one specific mutant we got in our screen. And this mutant is a ER chaperone NINA-A. So it's a chaperone, it's a ER resident cyclophilin, and the role of NINA-A is the proper folding of rhodopsin 1, which is the major protein of the photoreceptor cell. And what has been described in the 90s by Charles Zucker and Nancy Colley is that in the NINA mutant, you have accumulation of misfolded rhodopsin-1. So this mutant very likely activates the unfolded protein response. And that's actually what we checked first. So for that, we have a nice tool to measure ER stress unfolded protein response activation, which is a transgenic fly carrying a XBP protein fused to GFP. But when there is no ER stress, the EGFP is out of frame. When you have the splicing induced by the IRA1 gene, you have a frame shift, and the XBP becomes in frame with GFP. So it's easy to detect then GFP expression as a signature of UPR activation. So in flies expressing this transgenic construct, we can evaluate unfolded protein response, and you can see in the retina here, basal level of UPR activation, and in the mutant for NINA-A, you have increased activation, increased expression of GFP, which is also true here for a point mutation in the rhodopsin-1 gene. So now we ask whether in those mutant retina we have resistance or suppression of cell death. And the first thing I want to show you is quite intriguing, is that in the NINA-A mutant retina, the photoreceptors are actually pretty fine. They are all alive, even though they have some ER stress for their life. I mean, up to 3 weeks, there is no sign of degeneration by having this unfolded protein response. So I think this is the first example, or one of the first examples, of genetic preconditioning. So the fly is carrying a mutation which confer a preconditioning here. And what we can do is basically stimulate apoptosis, overexpressing apoptotic gene Reaper, which is a homolog of SMAC diablo. This is inducing a potent cell death here. And when you combine with NINA-A mutation, 1 copy or 2 copy, you have a suppression of cell death induced by the Reaper gene. We actually have shown in this paper as well that if you remove 1 dose of the rhodopsin-1 gene, you remove the stress, basically. This suppresses the protection induced by the NINA-A. So it's really due to misfolding that you have protection. Next, we ask, okay, so we know that we have ER stress, mild ER stress. This confer protection. What is the pathway behind this? What is the downstream pathway? So we knew that ER stress was tightly linked with autophagy. We heard this morning about autophagy in aging. And basically, we looked into whether this mild ER stress could cause autophagy, which is the garbage mechanism to eliminate defective mitochondria, aggregate, and so on. And for that, we use the GFP LC3 reporter, which is a good way to measure autophagic vacuoles. So we can see that in the NINA-A mutant, we don't see much of activation, but when we combine apoptotic signal with the ER stress, then you start to see punctuated vacuole appearance, and also the GFP LC3-2 band, which is a signature of autophagy. So we believe that cells which are preconditioned by ER stress, they are basically ready to activate autophagy when they are stimulated by apoptosis. Okay, so all that was fine. So we know that in Drosophila retina, there is neuroprotection by mild ER stress. So can we expand this data to other neurodegenerative disease? And in particular, we thought of Parkinson because not only we know that there is ER stress activation, but also because autophagy has a key role in this disease. So as you can see here, in the post-mortem analysis of brain of Parkinson disease patient, you have unfolded protein response, PERK activation, caspase-3 activation, and also sign of autophagy. So very briefly, Parkinson disease is the it's the most common neurodegenerative movement disorder. It's characterized by the loss of dopaminergic neurons, which are from the substantia nigra projecting into the striatum. And the selective loss of those dopaminergic neurons leads to severe locomotor impairment. One important signature is the aggregates due to accumulation of alpha-synuclein in Lewy body and also in Lewy neurites. So there are good models to study Parkinson, in particular in mouse, but also in Drosophila. Also, Drosophila doesn't have substantia nigra, neither striatum. It has dopaminergic neurons, which are important for movement. And we can study, for example, Parkinson's disease by expressing human alpha-synuclein in neuron of Drosophila. That's what we do in the lab. And when we here overexpress alpha-synuclein in the dopaminergic neuron, we see a progressive loss of dopaminergic neurons. You can actually stain them with tyrosine hydroxylase antibody. So there is age-dependent neuron loss in Drosophila. We have also age-dependent loss of locomotor function. We have this nice climbing assay where we can evaluate locomotion of flies, and flies overexpressing this alpha-synuclein have a progressive loss of locomotion. Furthermore, we can also evaluate synuclein aggregates. Here, when we overexpress huntingtin and alpha-synuclein, you can see those aggregates. So we can use this model as a Parkinson model, at least at the cellular level, to evaluate the death of the dopaminergic neurons. So can we ask whether ER preconditioning has a neuroprotective effect in this model? And for that, we actually used tunicamycin, which is an inhibitor of glycosylation, a known inducer of ER stress. Sorry. So tunicamycin can be added to the food of the Drosophila, and we can basically ask whether tunicamycin treatment, whether does it induce ER stress, and second, whether does it protect from alpha-synuclein expression in the fly. So first of all, when you feed flies with tunicamycin, it can go quickly to the brain, and you can evaluate unfolded protein response activation using the XBP-GFP here in all the neurons, or specifically expressed in dopaminergic neuron upon tunicamycin feeding. So it's a good system, so it goes right away to the brain. And when we do that, we basically, it gives some protection. So here is the climbing index. So when you overexpress alpha-synuclein, you see a progressive loss of locomotion, which is reduced when the flies have been fed with tunicamycin. So to go further, we decided to use another model of Parkinson's disease, which we spoke about a little bit about this earlier, is toxic chemical which actually affect the mitochondria, like paraquat, rotenone, which are known inducers of Parkinson's disease in a human. For example, farmers are intoxicated by paraquat, have been, at least in the past, or in some countries, they are still intoxicated by paraquat, and they develop later Parkinson's disease. And so we can use this model as well in the fly. So it's inhibitors of mitochondrial respiratory chain, and we can provide this to the fly and show that there is a loss of viability in flies exposed to paraquat, and if you pre-treat your flies with tunicamycin, there is a resistance to paraquat treatment. And using this model, what we can do is ask what are the important pathways in tunicamycin-mediated protection. And remind you, so we are basically focusing on the unfolded protein response, and what we can ask is whether, for example, the XBP pathway is important. So here, it's a similar experiment. When we treat flies with paraquat, or XBP mutant fly with paraquat, we can see that they die as control. And while the control flies treated with tunicamycin, they are protected, the XBP mutant flies, they are not protected by tunicamycin, indicating that XBP1 is required for tunicamycin-mediated protection. So then we tested another mutant. We can test several mutants. So we tested the PERK mutant as well. So PERK mutant, it's a bit different because PERK flies here in yellow, they are more sensitive than control. So it seems that to paraquat. So it seems that PERK activate is required for protection against paraquat. Maybe via a reducing translation like we spoke earlier. But interestingly, what you can see from the yellow to the red curve, PERK mutant flies can be protected by adding tunicamycin. So PERK flies are protected by tunicamycin, indicating that PERK is not required for this protection mediated by tunicamycin. So it's rather the XBP mutant. So another mutant that I would like to tell you, which is quite interesting, is a bit like the NINAE mutant earlier, which gives a genetic preconditioning, is the ERAD mutant pathway. And in the ERAD mutant pathway, what we noticed is that there is endogenous activation of the BIP. BIP is one of the UPR marker. So basically you block the ERAD, so you accumulate unfolded protein in the ER, and as a result, you can measure BIP activation. And those mutant flies, they're actually more resistant to paraquat. So again, it's an example of genetic preconditioning that we have here. So it's at the difference of the NINAE, which was restricted to the retina, here the HERP mutant is the whole body, which is resistant to paraquat here. So now, with the fly model, we basically wanted to conclude and go further to basically a mouse model to see whether ER stress genetic preconditioning via the ER could also be protective in a mouse model of Parkinson's disease. And for that, we used a 6-hydroxy dopamine model of Parkinson's disease, where you have injection of 6-hydroxy dopamine in the brain of the mouse, and this leads to selective loss of dopaminergic neuron in the substantia nigra. So in the protocol that we used, we can do a pre-injection, a prior injection of tunicamycin, intraperitoneal injection of tunicamycin, and ask whether this is protective against 6-hydroxydopamine. So the first thing we did, similarly as the fly experiment, is basically to test whether tunicamycin injection is sufficient to activate the unfolded protein response in the brain. So that was a key experiment before starting, and finding the right dose of tunicamycin not to being lethal, right? So we tested several doses, but basically the weak dose that we tested, 0.1 milligram per kilogram, was quite efficient at inducing XBP1 splicing in the whole brain. Importantly, SHOP, which is the pro-apoptotic, the bad guy, was not induced by those doses of tunicamycin. And we could also detect in the substantia nigra pars compacta BIP, which is also ER stress, but no SHOP activation. So basically we chose this dose, and basically we could apply our protocol, tunicamycin pretreatment, followed by 6-hydroxydopamine, and basically stain for dopaminergic neuron loss, and that's what we did. You can see here that basically 6-hydroxydopamine treatment induces about 40% dopaminergic neuron loss, and when the mouse has been pretreated with tunicamycin, there is a reduction of the cell loss. So that's a good news. So it seems that it works for the survival of dopaminergic neuron. And the last thing we did is basically assess whether this could be also rescuing a locomotor deficit induced by 6-hydroxydopamine. So the classical assay to evaluate locomotor deficit after 6-hydroxydopamine is a spinning assay, basically following apomorphine injection. So the mice are spinning, and what you can see that tunicamycin pretreatment reduce clearly the number of spin after 6-hydroxydopamine injection. So that was not only a rescue of dopaminergic neuron, but also a rescue of locomotor function by tunicamycin. And the last experiment that we did was to come back to the fly result. We showed that autophagy is important for neuroprotection mediated by the ER. Is it also true in the mice? And for that we could basically stain again with antibody against LC3, the marker of autophagic vacuoles, and we could see that when we combine 6-hydroxydopamine plus tunicamycin, we see increase of LC3 staining that we can quantify here. So again, cells which have been preconditioned by the ER, they are ready to stimulate autophagy when they receive toxic 6-hydroxydopamine treatment. To really verify that autophagy is important for the neuroprotection, we could use neuroblastoma cell line, again with the same protocol, 6-hydroxydopamine plus tunicamycin. We can see that there is a rescue of neuroblastoma cells with tunicamycin, and the rescue is lost when we treat a cell with scRNA against LC3, autophagy gene LC3. Similarly, with 3MA, the inhibitor of autophagy, similarly, we lose the protection with tunicamycin here. So together, it seems that autophagy is an important neuroprotective mechanism that is induced by ER preconditioning in our system. So to conclude, I would like to basically speak about this slide and what we know about the unfolded protein response, that overactivation of this pathway can lead to SHOP apoptotic pathway and cell death in some situations. It's true, for example, for high dose of tunicamycin or DTT treatment, it induces very strong death of the cells, in particular in the kidney. What we have shown in our work that at mild dose of ER stress, and we can induce it in different ways, either genetic preconditioning or tunicamycin, we are able to activate what we call ER hormesis. It's not a mito hormesis, it's an ER hormesis, in which there are different ways of inducing ER hormesis actually. So a classical ER chaperone mediated by the UPR, which will increase ER folding capacity. That would be the intuitive way of rescuing the cell. But also, I think what is novel is cellular protective mechanisms which are inducing by the unfolded protein response genes, and in particular we think that the IRE1 is important for protection and possibly inducing autophagy. Other groups have shown that antioxidant response can be induced by the unfolded protein response, and together I think what is interesting here is that the ER induces multiple protective pathway in the cell, and it's kind of combined therapy with one stimuli, right? And also, I think the interesting thing that we have here is that some of the UPR pathway are thought to be protective. That's what we see. The XBP1 is protective, while the PERK pathway is rather deleterious. So it's maybe not only the dose of stimuli which is important, but also the quality of the signal that we give. So we can maybe think of playing not only with the low dose, but also the type of molecules. So I think, for example, targeting specifically the IRE1 pathway with specific molecules could be actually very beneficial for rescuing neurons actually in a model of neurodegenerative disease. With that, I would like to thank people who did the work in the lab, current member with a PhD student, Marianne Cedru, and past member who have been involved in the project throughout the years, and as well as collaborators and fundings. Thank you very much.
Questions, Dr. Mollereau? All clear. You're welcome. That's good. So could you go back to your last diagram? Yeah. So I guess what I'm having trouble appreciating is when you're outside looking in at the system, as we all are, how is it that you're differentiating between, right? The easy one, right, is apoptosis versus everything else, okay? But most people think of the lower unfolded, I shouldn't say most people, I anyway, think of the lower level unfolded protein response as essentially being, as you say here, the increase of ER folding capacity. And everything else over here, I usually associate green books, etc. This right path with just that pathway, the ER chaperone normal processing.
Yeah.
How do you differentiate between those two?
Yeah.
Because, for instance, to say that a mutant is preconditioning, well, you could sort of argue like any retinal degeneration mutant is preconditioning for a disease that comes up later in life, because the human has that mutant in them the entire lifespan, but they don't start to see deficits until age 30 or whatever.
Yeah.
So you can say, well, there's all kinds of preconditioning pathways being invoked there. Is it just nomenclature, or?
I think yeah, the key thing is not only to show that there is increase of those pathways. That's what we see. We see increase of autophagy, and we also show increase of NRF2, for example, and other labs have shown NRF2. But the key experiment that you need to do is actually inactivate autophagy or inactivate the antioxidant response and see the loss of the protection. So you can see, okay, this pathway is contributing. And so these are experiments that we did in the fly and also in neuroblastoma. So when we inactivate the autophagy pathway, there is no more protection induced by tunicamycin, for example, so indicating that this pathway contributes in the protection induced by mild ER stress. But it doesn't exclude that I think all these pathways are working together for the protection. Yeah, so, yeah. So, yeah. Yeah, that's an important question to know whether all these pathways are activated at first and whether some of them are extinguished at some point. There is a nice paper from Peter Walter using the mouse, the rat model of retinitis pigmentosa. So they have this rhodopsin-1 mutation. So at the onset of the disease, so the cells are not dying, they are resistant. The pathway is ATF6, PERK, and IRE1, they are all activated. And after the disease progresses, later on there is a progressive loss of IRE1, ATF6, and only PERK, the deleterious PERK, remains active. So we think that the protective guys become lost while the toxic, let's say, PERK remains active. So I think it's not only the dose, the quantity, but also the quality of the response, which is important here.
And what is the specific switch that turns on those pathways from the ER stimulus?
Yeah, okay, so yeah, it's not represented here, but those pathways are inhibited by a chaperone, which is called BIP or GRP78. They are a normal condition inhibited when misfolded protein appear, BIP is recruited to the misfolded protein response, and its inhibition on the pathway is relieved and it's activated. So it really depends on the amount of misfolded protein in the ER, which then activate those pathways.
For the sake of time, I got a curtail of the questions. If you have additional questions or comments, we can just meet with Dr. Mollereau during our break. I'll break about 25 minutes. Thank you very much. It was a wonderful presentation.
Thank you. Thank you.